ladies and gentlemen, a very special edition of the Oppenheimer Ranch Project update. Tonight, we have the legend of legends, Paul Cottrell, literally the fastest growing YouTube channel on the planet, and my partner Leah Schaefer upstairs. Her video's back, so you can see her up in the top corner. I want to welcome both of these guests coming on the show because everyone is very busy, and Paul hasn't slept in 17 days. So, guys, welcome to the show. And how you guys doing?
Hi, I'm doing great. Thank you for having me.
And so
Paul's doing great despite a lack of sleep.
Yeah. All right. Let me just go over what we're going to be talking about tonight. The Wuhan coronavirus looks increasingly like a pandemic, experts say. Rapidly rising caseloads, alarming researchers, and we have just seen the 4th, 5th, 6th, 7th case of Wuhan in California just breaking minutes ago. The Johns Hopkins data was just updated in the last 30 minutes. 362 deaths, 487 recoveries, over 17,000 confirmed with the novel coronavirus. And other developments today, China completed the emergency coronavirus hospital, which will house up to 10,000 people. And that happened just in a period of less than a week. And here's the data. Health officials confirm 2nd, 3rd, and 4th cases of coronavirus in Bay Area and neighboring county. So as the virus spreads globally, U.S. travel restrictions go into effect tonight to combat the coronavirus spread. Guys, welcome to the show. Paul, you've been working nonstop on the topic. Can you give us a quick overview on the day's developments? Well, actually, we haven't talked in 2 days. So welcome to the show.
Thank you for having me. Basically, what's been happening in New York, which is where I'm living, is that there's actually 2 unconfirmed cases. They're being tested in the same hospital. One of the individuals actually is from Flushing, which is a station in the Queens borough, but basically in New York City. And the other one we think is actually a traveler from the Wuhan area. They don't live in New York. So we still don't know if they're tested positive. But even if they were tested positive, we don't know how many people they infected. There's a possibility that an outbreak has started in New York. But I don't receive any like special announcements, from the building managers or the city about what to do, what not to do. So we're just kind of like living our lives right now like we normally are. And that is a little scary because I think that's how this outbreak could start in high density areas. The first death outside of China took place, I believe it was yesterday, and that was in the Philippines. And it was I think it was a 44-year-old Asian male. And a lot of research has been, I'm very thankful for all the people that have come to my channel and that are helping a lot, and they're downloading lots of articles. And there is a very, very strong indication that this virus actually attacks males more than it attacks females. And the reason being is it's the ACE2 receptor. We talked about this, I think, in the last show, but basically the ACE2 receptor helps with controlling blood pressure. But because a lot of these enzymes are converted in the lung infections cause this blood pressure problem and people start to faint. But for males, there's also ACE2 receptors in our gonads, and I believe it's either the adrenal gland or in the kidney. I don't know for sure. I haven't fully read the research paper, but this falls in line with the 3-to-1 ratio that people have been claiming, that it's heavier on males than the females. it makes sense because of this ACE2 receptor, and knowing through published papers that go back all the way to 2010, where they were modifying the coronavirus, the SARS coronavirus, and the bat SARS-like virus to have a higher affinity towards the ACE2 inhibitor. And the reason why they were doing it was to understand which species of bats were the natural carrier. So this is just more evidence that this was bioengineered to understand the latency and the natural carrier.
That's a dirty
and which species
word.
It was.
Dirty word, Dr. Paul. We're not using that word on the show. Just kidding. Leah, you got to be happy to hear that you now have a 1 in 3 chance of getting it, while we are now 66.66% more likely to die than you.
Well, we're more likely to get it, not necessarily 60% to die, but
I'm
True
trying to be
that.
salacious.
Yeah, it's a little selfishly relieving, but it doesn't make me feel any better. I think that's interesting too, because I remember very specifically in the first study of what was like the first 41 people supposedly to contract this, that factor of males being more likely to contract the disease was in that first study. So it's interesting to see. So I was skeptical of it when I saw it then, but interesting to see that that's really turning out to be true.
And there's a rumor, and I don't know if it's true, and I don't know what the biological reason would be, but they're also saying that it may be ethnic targeting. That it may be more. This virus may actually be a stronger affinity for Asian males. And it could be just the way the data is. Where the outbreak was in Asia. It definitely is affecting more males. So obviously the data shows that there's more male Asians, and we just need a larger data set with more ethnic groups. But it would be extremely weird to have some more data points that points into ethnicity targeting. Because different ethnicities will have different biochemistries. And that's a fact. So, but what that biochemistry difference would be related to this disease is questionable. But the rumors are that there is some sort of ethnic targeting.
Yeah, we've
I don't think it's true.
Rumors,
I really don't think.
It's way too early.
I don't think it's true.
Yeah, and it's way too early even to speak.
But the same. Yeah, well, it was.
Yeah, there's no way we could possibly know that, simply because the geography is such that the people who are infected are in China first and foremost, and in Asia secondly. So there's absolutely no way to know that. Right.
But 6 days ago, the rumors about it targeting males and more males than females, the same logic held. So we have to be careful on this. But I do. I'll be careful.
Oh, by the way, Paul, Diamond just read that the man who died in the Philippines, it turns out he did not have 2019 NCOV. He died of AIDS.
Let me correct all that entire record, because I just went over that again, and so that is true, but. So the paper here, I'm showing it now, is the Chinese man who died Wednesday in Manila Hospital was negative for novel coronavirus. That was a 29-year-old Chinese man who died of AIDS. The 44-year-old Chinese man who died of novel coronavirus. So there are 2 people that died in Manila, and only one of them had the coronavirus. That is the correction.
Yeah, I was.
Oh, I see.
I was only referencing the 44-year-old.
Got it.
Now, Dr. Cottrell, let's real quick get into the paper that you and I and even Leah have been talking about. It came out 4 days ago, and it has since been withdrawn. The uncanny similarities of unique inserts in the 2019 novel coronavirus spike protein to HIV-1, GP120, and GAG. And so this has been pulled. Do you know about this?
Really?
Yes.
Really? I
Let
didn't know that it was pulled.
Let me read what it says.
Wow.
The paper has been withdrawn by its authors. They intend to revise it in response to comments received from the research community on their technical approach and their interpretation of the results.
Well, I ran I actually checked that sequence. All right, that G120 protein sequence from HIV, and also the GAG protein sequence. So GAG helps with assembling the capsid of HIV. The GP120 protein, I'm sorry, I'm trying to remember what that does. That helps with the affinity towards CD4 plus T cells. Okay, so I personally saw in BLAST the sequence that matches with the Wuhan strand that is left in the genome. It's left of the actual inserts that we've been talking about, that larger chunk on the right side of the genome, because we're talking about the 23,000 nucleotide position. It's left of that, and I saw it. Now, there's definitely HIV homology in the S protein in the Wuhan strand. There's no doubt about it. No doubt about it. Anyone could go into BLAST and look at the HIV, GP120, and see that there are many nucleotides that are matching unique features of the S protein, that as it's starting to fold, it creates this positive charge that is postulated to be to have that a higher affinity as it's locking into its receptor. The way proteins work is they fold. They're either hydrophobic or hydrophilic depending on charge, and they fold a certain way. And the researchers were saying that this was important for the docking onto a receptor. Okay, and this receptor, from the paper I just referenced and did a video on today about how they were taking coronavirus, the bat SARS like coronavirus, and HIV homology back in 2010. And people could just, people can, and I don't know if you provided me the paper. It must have been one of my subscribers that provided me the paper, but I'll have to research what paper it is. I'll send it to you. But it was published in 2010, telling people that they, outside of the bat, they were doing these assays to see which of the viruses, which strand of the virus that, with or without the HIV homology, would actually have a higher affinity, to understand if that particular species of bat could be a natural carrier. And so this research was going back all the way, quite far. So it's for them to pull the paper, there must be something that, because people need to understand that in the way the research world goes, it's all based on grants. That's how they fund their lab. So if they don't get a grant from either private philanthropy or from the government and toe the line, they are not, they're not going to get the money. So they probably pulled the paper because of methodology, but more that they don't want people to know that parts of HIV is in the S protein of the Wuhan strand, because most of the public is going to freak out. Most of the public is going to freak out and say, "Oh my God, I got HIV." No, you don't got HIV, but you do have a very virulent corona virus that needs to be treated. But the mass public is going to totally freak out when they hear that. But you and I, all three of us know, and we've seen multiple papers that this is real. This is not like, well, maybe it is, maybe it's not. There's papers saying we took that homology and was testing it for different reasons. So where are they going to, are they going to retract, 20 or 30 papers that go all the way back to 2010?
Probably not.
I mean, it's just like, you just got to,
But Leah, could you go over
the
the HIV, the AIDS protocol cocktail, and the mystery cure with our listeners?
Well, it's not exactly a mystery. There's a couple of different cocktails that are being used. So I just read an article about a woman in Thailand who was treated with an HIV cocktail, and in 48 hours, she tested negative for the virus. And that cocktail was oseltamivir, lopinavir, and ritonavir. And then in China, they're using lopinavir, ritonavir, and interferon. And in the U.S., that's usually known as a combo drug called Cottrell. It's those three things combined. So apparently it's looking like that can be very successful. And I think I read somewhere else that using an HIV cocktail like that was also effective for SARS. So, I mean, if there's a treatment option that brings somebody's viral load down to 0 in 2 days, that's very good news.
See, I think,
I
think that those drugs are actually helping with raising the white blood cell count. See, they're attacking, there's two problems that are going on with this virus. It's attacking the lung tissue, and it's also attacking the immune system. So I think this HIV cocktail that they're giving is to help boost the immune system, and that allows to have it fight the virus. I think the key part of this is that the immune system is turned off, and that's why it can cause so much havoc with the pneumonia and the scarring, the fibrotic scarring. By turning the immune system back on, you really have a fighting chance, and it's just a bad viral infection, but can be treated. That's why we've been saying that if you have an immune compromised person, this is probably really, really scary.
Yeah. By the way, Paul, I just learned today that typically in bacterial pneumonias, white blood cell counts go up, but when it's a viral or a mycoplasma pneumonia, it tends to go down or stay kind of at a normal level. So maybe that's why we're seeing lower white blood cell counts with this. However, when you combine that information with the potential for the insertion of, HIV sequences, I don't know.
Right.
I mean, that's kind of
right, because that GP, that glycoprotein 120 is specific for attachment to T cells. I mean, but obviously the glycoprotein 120 stands for the kilodalton, so it's the size of the molecule. But only pieces of that GP 120, amino acid sequence was inserted in to the Wuhan strand, just pieces of it to change the electro charge of the receptor to make it a little bit more. They have a stronger affinity, but it is that's a fact. The GP 120 does attack immune system.
I have a question for you. So something that's been bothering me is that every time I read about somebody's writing about the origins of this virus, and the answers are all so completely different. So the last time we talked, I mentioned there's a researcher named Zhengling Qi, and she's from the Wuhan Institute of Virology, and she's been working on coronavirus research for a while. And it was apparently in 2013, She found a coronavirus in a cave in Yunnan province. And there was an article published in Science magazine 2 days ago about this. And supposedly, one of these coronaviruses that she found in the cave with the bats, and this is also where people in the area, something like 3% of the people in the area had antibodies to these coronaviruses. But one of them, it's known as RATG13. And supposedly, it's 96.2% similar to NCOV that we're seeing now, versus, let's say, 79.5% similar to SARS. So I'm having a hard time in my mind reconciling, like we've talked about how this appears to be like two sequences of SARS viruses kind of like stitched together in some weird way, plus this HIV insertion. And I don't know how to reconcile in my mind all of these different descriptions about what this looks like and what it's similar to. How can it be 96.2% similar if we have these what look like bioengineered pieces of this virus?
Well, out of a 30,000 nucleotide sequence, RNA sequence, and the two big areas that have been inserted are about on the left side of the genome for the replicase is only 2,000. And the other side, I think, I'm going from memory now, maybe 600 nucleotides in insertion, maybe a little less. And the swapping of some nucleotides to make the HIV homology, because we're only talking about like 6 to 8, insertions for the HIV homology. The working hypothesis is that they took bat SARS-like virus that has an extra, and you probably have seen it, a little bit of extra code that is in this region that other SARS coronaviruses do not have. But some of the bat SARS do. So these bats that are coming out of the cave are probably have very similar homologies as the base strand that we're talking about, the bat SARS strand. Because remember, what we're saying is we're taking pieces of bat SARS and pieces of the SARS coronavirus and putting them together. And there's a big chunk are in all the coronaviruses, all the coronavirus families that are related to bat SARS. So, it wouldn't be surprising to go, because you got to remember what they were doing. They were engineering something that's very similar but supercharged that is out in nature. So if you go out in a cave and say, well, I found a bat that happens to have like coronavirus, and it's 90% similar to Wuhan, then therefore it must be an evolutionary a process that created the Wuhan strand. No, because the probability of these gigantic insertions, and when I did the probability, I gave it very generous probabilities for it to happen. But if you're talking about certain strands that have to be in the same cell and be able to do translocation and to be spit out of the cell and to be mixed in another cell with another strand, and then to be mixed again with HIV homology, and then be by a P4 weapons lab 20 miles away. The probability of that is like less than 0.01%. It's like closer to like 0.000001% chance of that happening. And I gave in each one of those, because it's a probability of and statements. So you have to times the probabilities together. But I gave, for every particular milestone, I gave it a 1 in 10 chance. That's very generous. It's like 1 in like 10,000 chance for that to happen. So I made it simple. 1 in 10 chance that for each one of these things could happen, and it could be by a weapons lab. just by chance, and just by chance gives you 0.01%, just by, and being generous. There's no way. And then we have a trail to follow with the literature that's going all the way back to 2010, in 2010.
Yeah.
That's showing that they were doing this. I understand why they were doing it. And it wasn't nefarious. They were trying to understand how the coronavirus works. It leaked out of the facility. And we have reports that facility has leaked out 2 or 3 other times. So,
SARS viruses.
Yeah. So, right. So what does that say? That says that the protocols in P4 Wuhan facility weren't safe. They did either bad hygiene, bad protocols, whatever it is, but it leaked out, and they didn't want to scare the public that, the great regime of communism could make a mistake. Because they have internal issues going on with President Xi. So I think it was just a bio accident. As some people in the Twitter sphere, it's like calling it China's Chernobyl. Okay, and what did Chernobyl happen? Chernobyl happened in the 80s, and it was a nuclear accident, and Russia was trying to hide it. But, we had, means to understand, we're catching these radioactive particles, when we're surveilling, this stuff, and people were getting sick mysteriously. And, they eventually had to, admit. Shortly after that, it was the fall of of communism in Russia. This is similar. It's not radioactive. And in some cases, it may be even worse. Because with radiation, it's more local, but it stays there for like 20,000 years. This is global, and it'll burn out after a year, but it may kill more people.
And very hard to avoid.
Yeah.
It's not
and
like
we don't
you can just leave.
Right. So I'm sticking. Even though technically it's still a working hypothesis, there's an awful lot of data points and probability that is saying, what? This is more than a hypothesis. That this is more leading to a theory.
So, Paul, the markets are opening now in China, and China has taken steps to limit short-selling activities in the country's financial markets. Right. It doesn't look like in after-hours trading that our markets are going to falter, but we closed last Friday down 660 points, 2.9%, I think. What are your thoughts on the markets now and China's move to limit short-selling and its effect on the global economy?
The last I looked, it was about 9% drop in the Asian markets, stock markets. Now, what I'm looking at is the yen relative to the USD. And that's trading at, I got my trading station on here. That's trading at about 108.5, roughly. All right. Once you start breaking like 107, 106, I would start worrying. But right now, it's stable. So either the central banks are intervening and making sure that there's nothing crazy that goes on in the currency market, which is very highly probable. And obviously you have to look at the gold market too, but or the Swiss franc to the USD. But for risk, for U.S. markets, I'm really looking at the yen. That's a great bellwether of risk in currency, which feeds to risk in equities, and the VIX. And the last I look on Friday, I think the VIX was around like 18 or 19, something like that. I don't remember. Maybe that was Thursday, but if you start seeing really long duration spikes in the VIX, like in the 20s, then you're going to start seeing repeated sell-offs. And what will happen is that in the U.S. market, if we start hearing confirmed cases and they start to accelerate, then we're going to start seeing a sell-off one day, maybe a little bit of a mean revert, but it doesn't get back to where it was the previous day, and then another sell-off, and then a little bit of a mean revert. So it's kind of like staircased, and it'll start selling this thing off. And I would have to do some technical analysis to give you a little bit more information on like support levels, but I would be worried once the 50-day moving average of the S&P 500 crosses over the 200-day moving average, then you're going to have extreme selling because of algorithmic trading, and it won't stop until some of these algorithms start saying, okay, too much selling, and then we're going to start to mean revert. But it wouldn't surprise me that we hear the what we call the cross of death, where that 50-day moving average actually crosses down on the 200-day moving average.
Leah, do you have any thoughts on the financial markets?
Sequence that was inserted called INS 1378, and he's saying that it's evidence of a recombination event in the gene, in the sequence. And I'm sort of asking this question because I don't really understand what he's talking about. But one of the things he says is that it has a strong similarity to a SARS spike protein, and that it also has a strong similarity to P shuttle SN vector. And he says that this was in use in the 1980s in China to create a more immunogenic coronavirus.
I mean, that's what I've been saying.
Does any of this make sense to you?
Yeah, I mean that S spike protein.
I didn't know if it was the same as what you've been saying.
That's the spike protein part of this is what I've been talking about. The S protein is the spike protein. Some people call it spike protein. Right. Some people call it the S protein. Now the P shuttle, I would have to do some more research on. I'm not familiar with it. I'd have to read the paper to understand what the reference of that is. But what we've been talking about for days now is this change in the spike protein. That's the very thing. Now there may be additional things that have been added to the genome that we aren't aware of that this paper may reveal. Because, all we know is, well, it's bad SARS-like coronavirus, and homology from HIV to make Wuhan, but is there something else that we don't know that's in it? Because it wasn't very clear when I originally scoured through the blast sequence. I didn't originally see, because it was so small of a sequence, I didn't see the HIV. We only saw it when we saw the paper that pointed us to looking for it to be able to say, reference this. Because the way BLAST works is that you pick a reference sequence. I can see you. No
okay.
issues.
You're back. You froze up and I lost my connection. Now you're back.
Okay.
So you were explaining the way BLAST works.
So the way BLAST works is that we have this sequence. So I took the Wuhan strand sequence that was originally given to us from China, and I searched for organisms that were similar. So it will rank based on percentage similar organisms. So I wanted to know that's the closest, that's like the fastest way to understand what was copy pasted in, and or how the virus evolved over time, right? Because you could see you could kind of see the evolution on some of these viruses when you look at the list, all right? But then all of a sudden there was a big jump. That's why they use the word novel. But that's kind of like a hidden term that's really saying, you know what? It's not zoonotic. It was created in the lab. Mm-hmm. It wasn't natural. Zoonotic meaning, in nature. Yeah. But this was lab created. And we didn't see that easily with the BLAST, which makes sense because remember, how did we know about the HIV sequence? It wasn't through the papers that go all the way back to 2010. It was that recent paper where they did the bioinformatics, because people can then focus on a little sequence. It's only like six nucleotide insert. And
you have to know.
So you had to know what piece of HIV to look for to compare it. So it's like finding a needle in a haystack. It's not as easy of I guess the point I'm making is the blast search is like this kind of like rough outline of possibilities of how it evolved. But little pieces, you have to really study that individually, with your reference sequence and other proteins that it might be similar to. Now, researchers in the lab would go, you know what? It has this kind of physiology, so it's similar to this physiology for this organism. Researchers that are working in the lab would have that detailed knowledge, but the average bioinformatic person would not have that detailed knowledge. But these papers give it the key to be able to find what they did. Now, by pulling the paper, we wouldn't know that. See, follow what I'm saying? Is that we would only know that there was an HIV homologous,
We're having some trouble with the connection. Clearly, we're getting deep into the subject. Maybe we should say our final farewells and take this back up another time. Are you there, Leah?
I'm here. I'm back. Yeah.
Okay.
I'm here.
Okay.
Connection's been going in and out.
Yeah, it's the Super Bowl.
Yeah, I mean, so I. Yeah, so I mean, so maybe that's what's causing the broadband issues. But the big point that I think the big take home here is that we would never have found the area on the genome that would have housed the HIV homology without that paper being released recently. And by taking it out of circulation, only a few people know it. Because it's going to be hard to find. They're never going to put that back up. I'd be shocked if they put that thing back up.
We'll see what happens. I mean,
but
corruption in science is not unusual.
Yeah, well,
for the reason you stated, grants.
Yeah, exactly. And if there was a methodology issue, why not keep it up on online, and then write another paper that has a better methodology and say this is to improve on the methodology that we once released. I mean, science it evolves. So they had a certain set of assumptions when they did the paper, and they have a new set of assumptions, and then they release a new paper that proves otherwise. But you don't remove it. So that's crazy. That's crazy. I mean, there's something.
I'll be interested to see how it comes out. If it does get republished with whatever changes, it'll be interesting to see what the difference is. But it's really unfortunate that happened.
Well,
and if they do change it, I hope that it comes out quickly.
BioRxiv, and I quote, "The copyright holder for this preprint is the author funder who has granted BioRxiv a license to display the preprint in perpetuity." And so this paper should be up for forever.
Yes.
And when I did search it, I got the paper again. Only difference was it said that it had been taken down.
Well, this is good because that site is a preprint site. That's what they publish. They're not publishing, heavily peer-reviewed stuff.
Well, I mean,
that's great because it'll stay.
These databases are to share working papers to collaborate with ongoing research to help fine tune before it goes to publication. I mean, they're designed to do that. I mean, it's a lot of these researchers do share their working papers.
So where do we go from here? We only have a few minutes left. The connection is getting cut. The borders are tightening. China is moving to control the markets so that they don't falter, and the infection rate is not slowing. We went from 12,000 yesterday to 17,000 today. We went from just under 300 deaths to 361 right now. And some of the leading virologists saying that they don't think this will peter out until April as a good target. What say you, Paul?
Right. No, I think that if once the outbreak starts, then it's about a month and a half to 2 month burnout. And then you'll have, a secondary and tertiary wave that, is attenuated usually. But when is the breakout outside of China? I'm working with the hypothesis that I believe that there will be a breakout in the United States, within 20 days from now, because of the this incubation period. But we're going to see much larger numbers in the United States, because this is not like Ebola. Ebola killed people quickly, and they were able to contain it very easily and control it. But once there is a breakout, it's going to overrun the medical system, and it'll spread, longer. We're really good at handling really infectious diseases, but only if there are a few, not if it's an epidemic. The system can't handle it. And I had a doctor that called me on my channel when I was live, saying this, saying that the system is taxed, and the nurses are worried. They're concerned.
Well,
that
let's talk about the fact that we have limited data on the asymptomatic nature of the spread of the virus, and if you can in fact spread it without any symptoms. They've been letting millions of people go past the checkpoint because they have no fever, and this might be ridiculous. So there may be, it's anyone's guess where it will go, and it may even mutate. What do you think, Leah?
I think that the idea that this can be contagious while asymptomatic is the absolutely scariest part of this. And frankly, I don't doubt that's the case. There's certainly been some papers that indicate that is happening. Certainly in China, researchers are saying this is happening. It took a while for the CDC to even be willing to consider that, which is pretty silly in my opinion. And then you couple that with things like, the mayor of New York City, Bill de Blasio, contacting surfaces, getting this from a contacted surface, and that's pretty scary. And that's the most challenging part of all of this. We can't really do anything about people who are asymptomatic unless we lock everybody down right now. The only way to deal with that, and the fact that could be a 14 days worth, it's anyone's guess what happens next.
I don't know.
I don't know. All we can do is hope for the best.
I don't know if you've noticed at your grocery store. I don't know how often you go to the grocery store, living in the mountains, but for us, it's starting to get, low on the food supplies. I mean, people are starting to feel that they need to prepare. So, it wouldn't surprise me that, people have this gut feeling it's coming. It's weird. It's not like SARS. SARS was not like this. Yeah.
No.
Well, yeah.
No. And I reacted the same way. SARS, MERS, swine flu. I kind of shrugged my shoulders. And for whatever reason, when this surfaced, I just had a bad feeling about it. A week ago, Paul, on Amazon, if you went to look to buy surgical masks or N95 or N100 masks, you could get them no problem with Amazon Prime, blah, blah, blah. And I just looked again the other night, and it's like they're basically unavailable. Yeah, people are prepping for this, and they are worried, and I think rightly so.
Mm-hmm. Yep.
What are you doing in Manhattan?
Well, I bought a lot of I bought a lot of food and hunkered down. No, well, I mean, like I was saying, before we went, live. The whole point was to, finish my master's and go to medical school and then buy a ranch and be a doctor in a rural area. But I'm stuck here with coronavirus.
Yeah.
Well, thank you again for coming on the show. We're going to be we're going to be keeping in touch as this rolls forward. Thank you for your time tonight. Go check out Paul's channel on YouTube. It is exploding. He's got over 13,000 subscribers. And 2 days ago we talked to him. He was just over 3,000 subscribers. So a very popular guy on YouTube. Check out his books on Amazon. Paul, do you want to? Is there anything you want to say to the listeners before we go?
I'm trying to start a kind of a live chat on YouTube. I did an experiment today with it. So just stay up to date and you can join in and call in and ask questions and share information. That's the whole point. So we can work as a community to try to figure out what's going on and how to prepare for this.
Awesome. Leah, final words? I can't hear you.
In this weird position where we don't know if the authorities are giving us information that's reliable, and then you couple that with a lot of rumors and probably weird theories on the part of the population, and it becomes very difficult for people to determine what they should and shouldn't be listening to. So having a central place to discuss what's likely and what's not, I think can be very helpful for people getting prepared and figuring out what exactly it is that they should do to care for themselves. Because we're not really getting all the information from the authorities. I'm quite convinced of that.
Well, thanks for your input. And we talked about two things on the show last night. Licorice root and red algae extract. Both are known to be excellent preventatives against SARS, which is sudden acute respiratory syndrome. In this case, the type of corona bat virus. Guys, thanks for coming on the show. It's been a pleasure. And we'll talk to you soon.
Thank you. Thank you.
Thank you, Paul.
You're welcome. Ciao.